Thanks everyone for coming. I guess I'll start off by saying that in the spirit of reconciliation, the Civil Society of Australia acknowledges the traditional custodians of country throughout Australia and the connections to land, sea and community. We pay our respects to elders past and present and extend that respect to all Aboriginal and Torres Strait Islander people present today. So welcome and thank you everyone for coming to the first branch meeting of 2021, at least for the SSA Canberra branch. My name is Francis. I'm the current president of the Canberra branch of the Stats Society of Australia. I guess just before we get started, a bit of housekeeping. Unless you are speaking, please try to ensure you mute your audio, just try and minimize the feedback. Video is entirely up to you, depending on whether you want to leave it on or off. Also, keep in mind that this talk is being recorded and we will make it available to SSA members afterwards. And that certainly is the plan. This first meeting in 2021 is actually a joint meeting between SSA Canberra and the Canberra chapter of RLADIES. And so it is a great delight to be able to have Dr. Gordana Popovic from UNSW Sydney speak to us about her research in this meeting. I guess what I'll do to start off is I'll give a bit of an introduction to SSA for those who aren't aware, and then I'll pass it either to Pauline or to Alice to talk a little bit about RLADIES before passing it over back to Gordana. So for all those who are not aware, the Statistical Society of Australia is the national body that represents statisticians and data scientists all across the country. And if you want to know more about all the events, workshops, meetings, and all that cool stuff that's taking place both nationally and in your local state and or territory, you can visit www.statsoc.org.au. I should mention in particular that this year SSA actually has two very exciting national online conferences that are taking place in the middle of the year. That is the Australian New Zealand Statistical Conference and the Early Career Statistician and Early Career and Student Statistician Conference. And again, you can find more about these conferences if you want to submit an abstract, participate, register, etc. You can find more information about those at www.statsoc.org.au. So I will now pass over to, I believe, most likely Alice, I think, who's going to say a little bit about RLADIES.
Sure, thanks very much, Francis. On behalf of RLADIES Canberra, I'd also like to extend a welcome to all members of RLADIES Canberra who have turned up to the meeting today. RLADIES is a worldwide organisation whose mission is to provide gender diversity in the R community. The Canberra chapter started in 2019, and we have recently hit 200 members, which I think is a fantastic milestone to have met. Our members are at all levels of R usage, from novice to highly experienced, and the chapter aims to run 3 or 4 events per year, sometimes in conjunction with partners such as the Statistical Society. So thank you to Francis for helping RLADIES Canberra to organise this meeting to kick off the year for both RLADIES Canberra and the Stats Society. RLADIES Canberra welcomes new members. You can join through our meetup site. It'd be great to have people who are interested in supporting that mission for gender diversity to join the group and join its activities. So thanks very much, Francis. I'll pass back to you.
Thank you very much, Alice. So our guest speaker today is Dr. Gordana Popovic, and Gordana is a statistical consultant and research fellow at UNSW Sydney with research interests in modelling and visualising multivariate discrete data with particular focus on ecology. And today she'll be talking about a topic that I think is relevant to a lot of us in data analysis, how to see the forest from the trees, removing pesky artefacts from discrete data for better visualisation. Over to you, Gordana.
Thank you, Francis. I have to apologise to everyone. I somehow got my wires crossed and I was pretty sure this was happening tomorrow. So this talk is still in very draft form, and hopefully it'll go okay. I was actually just bringing my mum home from hospital tonight, so she's home safe now, so we're all good. So I'm going to just talk a bit about visualisation and visualising discrete data. The field of visualisation is not my area of research. So if anybody wants to butt in and tell me I'm wrong about something, you're very welcome to do that. But I do know a little bit about discrete data and how to sort of treat discrete data. And so that also lends itself to visualisation. So we'll start with, oopsies. With just a little bit of like an motivating example. So I think most people have seen a graph like this. So, you do a linear model or some kind of model, and then you plot the residuals, and you might get like a graph on the left, which looks great. Everything is everywhere. We don't have any U shape, which you'll see in the third graph. So it kind of goes up and then down, and we don't have any fan shape, which you'll see in the middle graph. So the middle and the right graph are the ones to watch out for. The left is the good one. So my idea was to have a little quiz. Now, I believe you can use reactions. I think all of you have access to reactions. And I think there's a heart, and I think there is a celebration sort of thingamajig in the reaction. So we'll try and do a quiz with reactions instead of having a proper quiz due to my lack of organization. Okay, so what I want to know is if the left picture has the model fits better on the left, then I want a heart. If the model fits better on the right, then I want this little firecracker thing, okay? So here we go, let's give that a shot. So let me see if I can see your reactions. I got a couple of, so you think the left model fits better? Or the right model. So the right model should be a firecracker. The left, oh, I'm not getting very many reactions. Oh, I got a few firecrackers, good. A few firecrackers. And then if you don't know, just do any other emoji. So let's have everybody participate. If you don't know, just do any other emoji, any other reaction. I've got a crying one. Crying one's a good one because you don't know. Okay, so here we wanted the right one because the left one has this U shape, like it's down here and then it goes up and then it goes down. Can you see my mouse when I move it?
Yes, we can. Yep.
Awesome. Okay, so we got the one on the right. Okay, great. So what about this one? Do we want to go left for heart or right for firecracker? Got a right for firecracker, got some hearts, got lots of hearts, thank you. Thank you very much for participating. I know it's easy. Thank you. Oh, awesome. All right, what about this one? Remember, there's three choices. There's left for heart, right for firecracker, crying, or any other reaction if you don't know. I've got a couple of crying, I've got a left. Yep. Kind of a mix for this one. So from most of the others, I got the right answer, but here it's a mix. And the reason is this is from a generalized linear model, from a Poisson model. And it doesn't look like any of those graphs at the beginning. Whoops, it doesn't look like any of these because it's got these like artifacts that make them go in lines. And then you just can't really tell which one is the right one. I'm not going to tell you just yet which one's the right one here. I'm just going to do one more. Please participate for this last one. I think it's a bit of a palate cleanser after this one. So does the A model fit better or does the B model fit? So heart for the left and firecracker for the right. Heaps of firecrackers. Okay, excellent. So this is exactly the same model as this one. So the only difference between what I've done here and what I did there is what residuals I used to plot. So when I fit a GLM using just a GLM function in R, I've got, I can do, plot GLM. I've actually just plotted the residuals so you don't get a nice smooth line to make it a little bit harder for you. But I've just plotted the generic residuals that GLM gives you, and these are Pearson residuals. But actually, there's lots of kinds of residuals that you can plot. And so one type of residual that are quite good for discrete data are Dunsmythe residuals. And it's pretty easy to plot these in the Dharma package. So what you do is you simulate some residuals and then you plot them. And so these are from exactly the same model, which is the model up here. And you can see that it's ill specified. So there's a pretty strong U shape here, whereas here that it's there. Now that you know it's there, you can kind of see it, but it's really hard to focus on that pattern when all this other stuff is going on with the lines and the various things plotted on top of each other. So, so this is kind of a motivating example for what I want to talk about, which is mostly how to visualize discrete data without all these, what I'm going to call artifacts. I'm not actually sure. I couldn't figure out what they're actually called when you in the visualization world. Okay, so maybe I'll stop for questions. Let me find the chat. There's a chat. 9B. If anyone has questions, I'm keeping an eye on the chat. Francis, can you also keep an eye on the chat and then interrupt me if anyone has a question?
Will do.
Thank you. Okay. So Donemyth residuals. Okay, so I'm not sure where my notes are at this point, so we'll just have to hope for the best. Okay, so what we want when we are visualizing is we want the important patterns to stand out. So the things that we're looking for, in this case, we're looking for a U shape or a fan shape. These are the things that we need to find. And we don't want to have these sort of interfering artifacts that will mean that we can't see the things that we're there to see. And in statistics and when visualizing, we often expect, and by expect, I mean we're used to seeing continuous data, maybe Gaussian data with like nice symmetric data. And if everything is going well, like a normal, standard normal, unless we're actually interested in the mean or the variance, what we really would like to see is something very, very neat and normally distributed, especially when we're visualizing multivariate things. So what is the difference between those two residual types? Well, the Pearson residuals and other kinds of residuals, if the model's correct, they try to make the mean 0, so that's good. So everything's kind of above and below the mean, and the variance 1. And so these are both good things because that's what we expect from a linear model, mean 0 and variance 1. But in a linear model, the other thing that we get that we don't get in a generalized linear model with Pearson residuals is that they're normally distributed. So we don't get normally distributed residuals for the Pearson ones. And then for the Donemyth ones, we do. If the model's correct, what we get is something that's normally distributed. And that's exactly what we would get in a residual plot from an LM, which is the one that we're all most used to looking at. Okay, so here is what Donemyth residuals kind of are. So I'm going to assume that I've got some discrete data. So here I've counted the number of fish at a bunch of locations, and sometimes there's no fish, and then sometimes there's 4 fish, and so on. So this is just my observed data that I've plotted as sort of a histogram. But what I want to do is I want to model this data. So I say, well, there counts, so maybe it's a Poisson distribution, so I can fit the best Poisson distribution that I can find, and I can use, say, the GLM function to do that, and it'll give me this Poisson distribution. So you see it's pretty close to the observed data. It will have the same mean, and it looks pretty good as a distribution. So now I've got some raw data, I've got a distribution. How well did I do fitting this distribution? Well, if I want to know how well I did, I can always plot the residuals. Here is the DARMA residuals diagnostics. This is actually what they do. I think it's a little bit over the top, but you can see this is the normal quantile plot, and that's looking pretty good. So these residuals are okay. So, whoops. So what we do, okay, so we've got the observed data. We've fit a model to get an estimated density in green. And then once we have that, we can calculate the estimated distribution. So the distribution is just cumulative. So here it's the probability that it's less than or equal to 0, which is the same as the probability that's equal to 0. And here it's the probability that's less than or equal to 1, which is the probability of 0 plus the probability of 1. So you can see that if you stack this green one on top of this other green one here, you'll get up to here, and so on. You stack both of these here and you get there. So this black line is just our cumulative distribution function from our estimated density, which is in this case a poisson. Yes, it was. Someone asked histograms for discrete count data. I wonder, Alan, what you mean by that question. Okay.
Sorry, Gordana. Usually histograms are for continuous data to represent crude representation of a density, and spikes would represent the probabilities in a discrete distribution.
Yes, you're probably right. They are separated technically, as you can see. So they're not quite histograms, but I may have said histogram. But thank you. Thanks, Alan. Okay, so here I have my cumulative distribution function. So are we following up until this point? Who knows? But up to this point, all I've done is I fit the data to a GLM, and then I have used that to calculate the cumulative distribution function. And so this is at this point, I fit my model. I want to do some residuals. I want to plot some residuals, for example. So one way to do that is to use this cumulative distribution function. And so what I do is, for say one of my observations is 2, then I go to the line of 2. So the probability that I observed less than or equal to 2 and less than 2 and less than or equal to 2. So between these two probabilities. And what I do is I simulate a uniform number here, and then that will give me a uniform number on the blue area overall will give me a uniform on the whole red line. And this is just the probability integral transform. So, so I can do that. So when I have a 2, my residual for 2 is now going to be whatever that turns out to be. But I might have more than one 2, right? So I need a different 2 that I observed will get a different residual. So that might be this number here. So these are now uniformly distributed. And if I want something that's Gaussian distributed, then I can go backwards through the Gaussian distribution to get 2 values that are Gaussian distributed. And this is how I end up with my Donemyth residuals. Does someone want to ask a question about that? Is that super unclear? Probably. I'm not even sure what comes next. Oh yeah, okay. So far I've just introduced Donemyth residuals as kind of a neat way to do residual plots for GLMs, which have some good properties. So not only do they have mean 0 and variance 1, but they're also normally distributed, which makes them look a lot more like the residuals from a linear model with Gaussian data, which makes it easier for us to interpret the things that we need to find, like the U shape or the fan shape. So that's one thing. But we're here to, what I really want to talk about is how to visualize multivariate data. And so what we're interested in seeing when we're visualizing multivariate data are relationships between variables. So maybe we want to see if things are correlated or interacting with one another. Also relationships between observations, so maybe clustering or outliers or covariate relationships. And if we have discrete data, there are a couple of things that can obscure these things that we're interested in seeing, mostly the mean variance relationship. So if we have counts, for example, so when the counts are on, when you're expecting maybe 3, when you go counting, you might get a 2 and a 1 and a 0 and a 4 and a 5. So they're all pretty close to 3, like within 2 or 3 of 3. But when you're expecting to find 1,000, you might get 980 or 1,070. So the spread that you kind of would expect is a lot larger. So that's just a normal thing with counts, for example. So the variance for small counts is much smaller than the variance for large counts. Also, there's the discreteness, which was the main problem that we were having in the residual plot, where all the dots are kind of lined up together. That's because the data is discrete and lots of those are the same. And I'm going to skip that because I don't know what we're doing with that. Okay, so I might just take a second. Francis, when do I stop talking?
Probably around about 7.
Okay. Okay, so I'm not altogether sure what comes next because I'm only halfway through putting this together. So hopefully there's some stuff in there that I want to talk about. The first one I'm going to look at is a continuous some continuous variables. Sorry, a data set with continuous variables, but more than once. So a few different continuous variables, and I'm just chosen the penguins data set because it's fun. And it's just some measurements from some penguins in Antarctica, and often used for visualization these days and for other things. So I guess one way, the first thing that I often do if I don't have too many variables to visualize is I do a pairs plot because it's kind of a convenient thing to do. But like if you use the pairs function in R, it's a little bit not as fun, not as informative as you might want it to be, and not as neat. So I quite like this sort of GG pairs function, which lets you do, you know, colors and kind of fits in with the whole GG universe. So here we've got a few variables. So the length of the bill, the depth of the bill, the flipper length, mass, and some isotopes. And in the colors are the 3 species of the penguin, so the chinstrap, the gentoo, and the Adele. I actually don't know which way around they are. And so what can we glean from this? Lots and lots of stuff, right? So we can see that they're pretty well separated in some of the variables. So for example, the red ones are separated by bill length, so they have much shorter bills. But if you look at bill length together with flipper length, then you can see that all 3 sort of species of the penguins are quite separated. They also all have about the same spread, so then they're pretty well sort of equally sized, sort of clustery things. So that's interesting, and regardless of what plot you look at, that's sort of true. And so that's really it for the penguins. Basically, you can get quite a lot out of this plot because what we've got is some continuous data that has, you know, a smallish number of dimensions. So we've got 6 dimensions, and we can see quite a lot. If the other thing we can do is, I've recently gotten into tours, and tours are really great. There's a tour package. I haven't got the code up because I will put up some code. But a tour is a projection. So here in the pairwise plots, what we're doing is we're looking at bill length against bill depth from that side, and then bill length against flipper length in this one. So we're kind of looking at it from a particular side in this 6-dimensional space. But only one of the sort of at 90 degrees sides. Whereas when you do a tour, you can kind of turn in any direction. So this is a tour of these data. And it's a really fun thing to do. Oh, I did leave the code. So there's like the how to do a tour. So I can move this around and I can see sort of the same patterns I was seeing earlier. They're quite well separated depending on how you turn it. and the clouds are kind of the same sort of size. So the spread in each of the dimensions is approximately the same for all the different penguins. So that's really neat. And I definitely recommend tours as a thing to get into. But what if we have discrete data? So I've left this slide here as a blank slide. The reason is it's very hard to get a cute picture of a hunting spider. I did try, but if you are not, if you're a bit arachnophobic, just look away for a moment. Here are some of my spiders in this data set. So these are just, this data set is, there are a bunch of spiders that were collected along with some environmental variables. And so basically they were collected using pit traps. So people dug a hole in the ground and put like a Coke bottle in there, and then the spiders fall in, and eventually somebody comes along and counts how many of each of these spiders. And they did this at about 28 spots, and altogether they found 12 species of spiders. So and then there's some environmental variables like how many, like what proportion of the ground is covered in leaves, that kind of thing. Okay, so here if I do a pairwise plot of this data, this is what I get. Okay, so what's the difference between this and the penguins? One of them is that it's not nearly as satisfying a plot. It's very hard to see anything. So some of the reasons for that are, this data is very skewed. There's lots of small counts for all of the different species. So these are the different species. These are just 6 of the species. Lots of small counts, but then a few large counts. And the colors are here by whether or not there were leaves on the ground in that spot. So you can see maybe that this spider does, leaves are red. The spots with leaves, I think, are red. And so you can see that when there are leaves, this spider tends, is more abundant maybe than when there are no leaves. But it's really hard to see a pattern between the variables, like are they correlated? What's going on? Maybe there's like an increasing relationship. So these two are maybe positively correlated. It's hard to say. There's lots going on, but really hard to do anything with that. So what can we do about this problem? Usually you would transform the data. So you might go, well, it's discrete, it's counts, so maybe I'll do a log transformation or a square root transformation or some other transformation, box-cox. And we do that for sort of similar reasons as you do in the residual plot. We want to stabilize the variance. We want to remove the skewness, make things more symmetric. And so I'm calling this kind of the Pearson residual solution to this, to the problems we're having with visualizing this discrete data. And transformations can work pretty well on large counts. They pretty much work quite well on large counts. They're less good on small counts when you have lots of zeros, because all the zeros will end up exactly the same regardless of what you do. When you have ordinal data or binary, all of these it's hard to know what transformation to use. Binary, it's not clear what you can do at all. And you also can't, sometimes you can sort of have an idea of what transformation is correct, maybe with the box-cox, but generally speaking, you're kind of guessing at what transformation would be good. So one way you could do something with this is by using Donemyth residuals. And so Donemyth residuals use a model. So you fit a GLM in order to come up with the distribution and therefore the Donemyth residuals. So what you're doing is you're modeling the underlying distribution of the data. And then using that model to create residuals that are going to be standard normal, assuming that your model fits well. So you can think of it kind of as a more principled transformation of the data. And we can check that we use the correct distribution. So that's what we can do a residual plot and see that we did the right thing. And it also removes discreteness, which I like. So, we saw earlier that separate observations of like two spiders. So if we have 2 spiders in one spot and 2 spiders in another spot, you're actually going to get a slightly different residual for both of those. So it kind of jitters your data, but it does this in a very principled way within the bounds of where that jitter should really be. And so I think it tends to be a good thing, I think. And so, yeah, what does modeling mean? I just mean we're modeling the distribution. So we could do Poisson or whatever with a GLM. So here is, if I, you can do Duns-Smythe residuals in a bunch of ways. This is the package Ecocopula, which I wrote and Francis has contributed to, and some people in Michelle in my lab. And so there's this function called stacked SDM, which is kind of it's named for ecologists, but you can think of it as like multiple GLMs. So what I'm doing is I'm fitting a GLM to each of the spiders. And they're going to be negative binomial rather than Poisson, because that's what makes sense for these spiders. And then when I call the residuals function, that's just going to generate some Duns-Smythe residuals by default. So that's what this does. And then I'm just going to plot those with a pairs plot. And so now this is my plot of the Duns-Smythe residuals. So first of all, I'm much more pleased with this plot than I was with the other one. It doesn't frustrate me as much. And I can sort of start to look for patterns. So now I can pretty clearly see that the red ones are on the right here and the blue ones are on the left. And there's a bit of overlap with this spider here. And you can see sort of quite a good separation between the reds and the blues when you look at both the trucker spider and the parapole. These are just, these are actually not the spider names. These are kind of combinations of the genus and the species. But you can see a fair bit of structure in the data that we really couldn't see before. Can I just switch back? I guess now that I know it's there, I can sort of see it. But I definitely wouldn't have been obvious to me that there's like a pretty good separating line between the reds and the blues on that one. So so yeah, essentially that's what we end up with when we do Duns-Smythe residuals for these. We can also do a tour. So this is a tour of the raw data, so not transformed, not Duns-Smythed. So what am I looking at? So I guess what I would say for this data is I feel like there's like a cluster here of reds that are quite close together, and then there's a few that are sort of far away. So there are kind of 2 types of reds. If you know, if I'm looking at it, that's what sort of jumps out at me. They might, like, I might call this a cluster of reds, and then the blues are sort of more equally spread out, whereas the reds, like, there's one cluster and then there's a few outliers from that. Maybe that's what I would say. I don't know what you would say. That's what it kind of looks like to me. But if I do the Duns-Smythe residuals, well, the reason that those reds were all together is because they were all very small counts. And so they were all stuck together, basically. They were all exactly the same. And the reason that they were close together was because their variance was low. But we're not interested in looking at the variance within each species. We want to know how the species are related to one another. So we're visualizing multivariate data here. And so when we look at the Duns-Smythe residuals, we get quite a different picture. So you can still identify the ones that were all kind of close together, which is this cloud there. Where is it? It's that cloud over here. But we can see now that actually the reason that they were stuck together wasn't for any, like, that they were really clustered together. They were just small counts and therefore less variable than large counts tend to be. And so that's just a normal thing for counts to be, and not actually an interesting thing about this particular data set. And now that we know that, we can sort of see that, well, I think that you can, I think the spread of the blues is similar to the spread of the reds now that we're sort of here. So I think it's kind of, it's not much to be said about it, but yes, you can definitely find a projection where they're quite separated from one another. Maybe like that. But there are some sites with leaves that are more similar to the sites without leaves than the other sites with leaves. Okay. Yeah. Okay, so people have noticed this before, of course. And one paper that talks about this is David's paper. And it talks about it in the context of the MDS, so multidimensional scaling, but it's also true in this context, I think. So what this paper says is you end up sort of, if you don't do the right transformation, or you don't transform at all, you end up confounding the dispersion and location effects. So whether or not things are close together because they're a cluster or because they are just have small variance because they're small counts. And then also variables with small counts tend to be overlooked because the ones with small counts will have small variances, and then you won't be able to see the pattern in those ones. And you'll just be looking at the variables with large counts. Okay, so all of that I've just talked about would have been very highly referenced probably had I finished my talk properly. Oh, there's the tour method.
There was a question in the chat. Oh, sorry. Do you still have the chat up, or?
Let me just see. Oh no, I can't see the chat anymore.
Oh, okay, okay. So James Murray asks, does the tour method project just a 2D space?
Oh yeah. Well, did I answer that, or did I cut out before that?
You were cut off. Sorry.
Oh, okay. All right. Because I was going on about that for quite a while. Okay. So the tour method can project to different spaces. So this is like a, so here I've got 6-dimensional space. So exactly the same as that pairs plot I had earlier. So this is all the dimensions that are in here somewhere. And then it's projected onto 2D space so I can plot it as a scatter plot. Which makes sense for a talk. But you can also project it onto higher dimensional spaces if you want to. I find it hard to look at those. I prefer a 2D projection than a 3D one. But yeah, you can definitely do that. It feels like it might be a little bit 3D, but it's actually not. This is all just 2D. Yeah, does that sort of answer your question?
Yeah, thanks, Gordana.
No worries. Any other questions?
No, not at this point.
Okie dokes. Okay, so so I don't actually work that much on just normal visualization stuff, except for this little bit here where we're doing Donemyth residuals and tours together. Francis and Diane Cook and David are working. We're working on making that a little bit easier to do. So look out for that. But most of my work is actually it's related, but it's not quite in the same space. So sometimes you don't like, and other ways of looking at multidimensional data is to somehow collapse it or summarize the way that the variables are related. So you might think about PCA or factor analysis as a way to sort of reduce the dimension of the space that you're looking at. Or Gaussian graphical models, which are, I'll show you in a minute. There are ways to, so you can plot a graph which has sort of connections between the species, whether or not they're correlated. Also clustering methods and various other things. And so all of these methods, at least the sort of baseline version of them, expect Gaussian data. So if you do a factor analysis using in R, it works best. It's sort of designed for Gaussian data. Gaussian graphical models definitely designed for Gaussian data. Lots of clustering methods work best with Gaussian data. And so they don't necessarily work really well for discrete data for all the same reasons that we've been talking about. So what I tend to work on are actually copulas. So this is a model for multivariate data. So we have these, say, 6 species of spiders. We want to model them jointly. What can we do to model them jointly? Well, there's a couple of things you can do. You can do hierarchical models like using GLMMTNB or LME4 or various other things. You can use generalized estimating equations, but you can also use copulas, which outside of finance are very rarely used. But I like them because I like Duns-Smythe residuals, and actually Gaussian copulas are intuitively quite similar to Duns-Smythe residuals. Basically a incorrect way to describe Gaussian copulas, but a way that gives you a good intuition about what they are is the following. So it's not quite correct, but it makes it easier to understand. So the way that a copula works is, say you have a bunch of species, you calculate some Duns-Smythe residuals for each one. So you're fitting a GLM to each one, you do Duns-Smythe residuals, and you know that these are going to be, each of them standard normal distributed, because assuming that the model fits well, that's what the theory tells us. Then you go one step further and you assume that they're jointly normally distributed. So this might not be the case, probably isn't, but you would make that assumption. And then you can do factor analysis on those Duns-Smythe residuals. Now, this is not exactly what copulas do, but it's kind of what they do. And it's a similar thing to what they do. And so they're quite sort of similar to these Duns-Smythe residuals. Okay, who likes math? I'm going to skip that. This is how copulas are related to Duns-Smythe residuals, but I'm just not going to do that. But basically, this allows us to do things like factor analysis type models, but for discrete data using copulas. So here I've got the spider data, and here are the sites. This is basically a factor analysis with 2 factors. So this is the first factor, this is the second factor. And so some of the species are, so for example, these sites here tend to have more of these 2 species, and they tend to be more similar in terms of their species composition and like the kind of species that tend to hang out there than they are to these ones. So that's kind of a cool way to also visualize data, but it's based on a model. It's based on a projection of the data. And then you can also do graphical modeling. Graphical models are really fun. Basically, they tell you relationships. Yes. So James asked, is this related to PCA, principal component analysis? It's almost the same thing as PCA, James. Quite similar. But PCA will also work best with Gaussian data. So if you wanted to do, strictly speaking, PCA, you could also do it with Duns-Smythe residuals and get something maybe a little bit more interesting. So the graphical models, this is a graphical model of my spider species. So what this is, it's kind of a network of the species. It's estimated from the data. And so what it says is that this spider here, aloe, let's go call it aloe, is related to PAR. So these 2 spiders are related even after you take into account all the other species. These are still correlated, so they have a partial correlation between them. So that's how you interpret this. Whereas this one here and this one there, so Para, Mont, and Trucker, they don't seem to actually have any relationship between them. What's your condition on all the other species? So once you control for all the other species in the data, that these are no longer correlated. So they're kind of, their residuals are no longer correlated. So it's a kind of a cool way to visualize data. When we have so little data as we have here, so there's like 28 sites and 12 species, so it's not a great amount of data and it's discrete. You take this definitely with a grain of salt. It's not going to give you like hypothesis tests or anything, but it's a neat way to sort of visualize what might be happening between the species. So I think I'll probably leave it there. I think that's all I really have, do I? Yeah, I don't think I finished the rest.
Cool. Okay, thanks, Gordana. I think you did an outstanding job, particularly given you're sort of doing this half on the fly, and particularly given that there was a bit of a technical issue on the way. Thanks for everyone for tolerating sort of Zoom's technical issues along the way. I guess if anybody wants to ask a question to Gordana, you can do it through the chat, or you can also use the